This will just be a continuation of our discussion on how plant hormones promote seed germination. In this lecture, we will focus our attention on the remaining three major classes of hormones, which include cytokinins, auxins, and ethylene. Cytokinin was first discovered in the DNA of the herring fish. The hormone was reported to promote cell division, and by the time of its discovery, it wasn't even named cytokinin, but kinetin. Soon after, a substance that had the same biological effect as kinetin was found in plants. It stimulated plant cells to divide when cultured with auxin, another plant hormone. The substance was named cytokinin, and it is involved in cell division and in the making of new plant organs like a root or a shoot. Cytokinins are produced in the root apical meristem and travel upward, hitching a ride with water and traveling up the stem through the xylem. The movement of cytokinins is passive, meaning it does not require any energy. Cytokinins are like the fountain of youth in plants. They delay senescence or the natural aging process that leads to death in plants, and cytokinins do this by encouraging cells to divide. In rice, the accumulation of cytokinin results in increased grain number. In this panel, we have the rice cultivars koshikari and habataki. These two cultivars differ significantly in terms of yield, as you can see in this panel here. This is the panicle of koshikari, and this is the panicle of habataki. If we look at this graph, we can clearly see that habataki produces almost twice the yield of koshikari. Habatak here is abbreviated as HA, and koshikari is abbreviated as KO. Now, the ability of habataki to produce more than double the yield of koshikari is because of a mutation in its GN1A gene. GN1A in rice codes for cytokinin oxidase dehydrogenase that degrades cytokinin. Habataki has a defective GN1A, which means it cannot degrade cytokinin. If it cannot degrade cytokinin, then it means cytokinin accumulates in the plant, resulting in the higher grain number. Cytokinin is active in all stages of seed germination. It can enhance germination of seeds by alleviating the effects of abiotic stresses such as salinity, drought, and even heavy metal contamination. Seeds that have specific requirements for low temperature or light can be induced to germinate with cytokinin treatment. A good example of seeds with chilling requirement are Leucadendron daphnoides and Protea compacta, while example of seeds with light requirement are Rumex obtusifolius, lettuce, and celery. Without meeting these two environmental requirements, seeds of this plant species will remain dormant. But as I said, this dormancy can be broken down by cytokinin treatment. This slide illustrates how cytokinin affects germination in Arabidopsis seeds. We have here two Arabidopsis genotypes. One is Colombia, abbreviated as COL, and we have GIM1, which is a mutant with reduced sensitivity to abscisic acid or ABA. In the experiment, seeds were grown in plain growth media or control, in a growth media supplemented with ABA, or in a growth media supplemented with ABA plus estradiol. Estradiol is abbreviated here as E. This is a compound that induces the expression of a gene encoding isopentyl transferase, which is responsible for the production of cytokinin. Now, if we examine this panel, we can see that both Colombia and GIM1 germinated in the plain media. But when treated with ABA, which is a germination inhibitor, we can see that germination slowed down. When treated with ABA plus estradiol, we can see positive effects on the germination of both Colombia and GIM1 as indicated by the green cotyledons here and also in this panel. So why do we see a more pronounced positive effect in GIM1 but not in Colombia? It's because GIM1 has lower sensitivity to ABA compared to Colombia. In the presence of estradiol, which induces the production of cytokinin, GIM1 was able to overcome the inhibitory effects of ABA. Now, the right panel shows the effects of increasing ABA content, applied alone or in combination with estradiol, on GIM1 and Colombia germination. You can see from the overall trend that GIM1, treated with ABA and estradiol, was able to germinate better even at the highest ABA concentration as indicated by the line with a triangle. Again, this is because GIM1 has reduced sensitivity to ABA. Here's another example that illustrates how cytokinin affects germination in Arabidopsis seeds. We have here again the two Arabidopsis genotypes Colombia, abbreviated as COL, and GIM1, which is a mutant with reduced sensitivity to ABA. In this experiment, seeds were grown in a plain growth media, or control, in a growth media supplemented with ABA, which is a known germination inhibitor, or in a growth media supplemented with ABA and 6-benzylamminopurine, or 6-BA. 6-BA is a synthetic cytokinin. Now, if we examine this panel, we can see that both Colombia and GIM1 germinated in the plain media. But when treated with ABA, which is a germination inhibitor, we can see that germination slowed down or was even inhibited in some seeds. When treated with ABA plus 6-BA, we can see positive effects on the germination of both GIM1 and Colombia. This is Colombia and GIM1, although the positive effects were more pronounced in GIM1. The positive effects are indicated by the presence of the green cotyledons in both Colombia and GIM1. So why are we seeing more pronounced effects of 6-BA in GIM1? Again, this is because GIM1 has lower sensitivity to ABA compared to Colombia. Overall, the results of the study show that number 1, cytokinin is a positive regulator of germination, and number 2, cytokinin can reduce the inhibitory effects of ABA in germinating seeds. Then we have the auxins. Auxins were first discovered in the late 1920s, and it was the first of the 5 major types of plant hormones to be studied. Auxin is involved in plant growth and development, cell growth and expansion, and formation of vascular tissues. It is produced primarily in parts of the plant that are actively growing, like the stem, specifically the tip of the stem or shoot meristem. Auxin is transported in one direction in a plant, which is downwards, from the top to the bottom. The transport of auxin from the tip of the stem to the root is an active process and therefore requires energy. It is the only plant hormone known to do this. Therefore, the concentration of auxin is going to be highest at the top of the plant and decreases as you get closer to the roots. Let's take a look at an actual example of how auxins affect plant development through the process of phototropism. Have you ever noticed how a plant in your windowsill bends towards the sunlight? This has to do with the auxin in the stem. Auxin, as I said, is involved in making cells expand. So if you put a plant outside, just like in this panel here, free of any shading, the plant will just grow upward. But when you put a plant in the windowsill, the auxins are going to move to the shaded side of the plant and cause those cells to grow longer, while the cells on the sunny side of the plant stay the same size. That will cause the plant to bend to one side toward the sun. Because auxins are produced in the tip of the shoot, it controls the overall shape of the plant and helps keep the primary stem of a plant as the leader. This control is also known as apical dominance. In this panel, we have a plant with a single stem. But if you cut the shoot, what will happen? The plant loses its apical dominance and start producing new lateral shoots. That is because auxin maintains apical dominance and by doing so prevents lots of lateral buds and branches from growing on the side of the stem. When you prune the primary stem of a plant, the source of the auxin is removed, then no single stem is dominant anymore. Apical dominance is essentially removed. With Christmas trees, apical dominance is very important to maintain the perfect tree shape, like what you see here. Cutting off the main shoot will remove the source of auxin and will result in the sprouting of new lateral shoots or branches. This is an example of the effects of auxin in the growth of corn and its wild relative teosinte. This is a corn plant that produces auxin. We don't see any branching. On the right is a picture of teosinte that is defective in auxin synthesis. In this mutant, we don't see any apical dominance or dominance of a main stem. Instead, what we have here is a plant with several branches. At least we have here three branches, one, two, and three. During germination, auxins are not really necessary, but its interactions and crosstalk with other plant growth regulators like gibberellins, ethylene, and ABA influence the germination of the seed. In general, higher concentrations of auxins can inhibit germination, but lower concentrations may promote or may not have any effects on the germination process. This slide illustrates the effects of exogenous treatment of seeds with auxin. We have here soybean seeds that were germinated in water, which serves as our control setup, and seeds treated with indole acetic acid or IAA. IAA is a well-known auxin. As you can see, IAA treatment slowed down the germination of soybean seeds as indicated by the shorter radical after 18 hours of germination compared to the radical of that of the control group. These graphs below also show the effects of IAA on the actual length of the radicle and the fresh weight of seeds. As you can see, IAA treatment resulted in shorter radicals and lower fresh weight of seeds as indicated by the orange bars in both histograms. This slide also illustrates the effects of IAA on the germination of two soybean cultivars ND12 and C100. Seeds of both cultivars were germinated on 2 layers of filter paper in Petri dishes. Seeds were treated with water as control or 1 micromolar IAA. All the seeds were germinated under dark conditions. As you can see from these pictures, as well as from the graphs in the right, treatment with IAA reduced seed germination in both cultivars. See how the radicals are protruding in ND12 and C103 that were germinated in water only, but not so much in seeds treated with IAA, as you can see from these panels here. And when we look at the graphs, we can see that germination of seeds of both cultivars after 36 hours was lower with IAA treatment as indicated by the orange line here and also here, compared to the control as indicated by the blue line here and also here. Last but not least, we have ethylene. Have you ever noticed that if you put a really ripe yellow banana right next to a bunch of green bananas, the unripe bananas will ripen and turn yellow much faster? How does that happen? Well, the yellow banana is actually communicating with the green bananas using a hormone called ethylene. Ethylene is a plant hormone that affects ripening and rotting in plants. It is the only plant hormone that exists as a gas. It is produced in any part of the plant and diffuses through the plant's tissue outside the plant and travels through the air to affect a different plant. Ethylene was first discovered by tomato farmers who noticed something weird happening with their crops. So back in the day, many farmers used kerosene heaters in their greenhouses to warm the air so that they could grow tomatoes during the winter. But with the advent of electricity, some farmers switched to electric heaters. They soon noticed that their tomatoes were not ready to be harvested at the same time the way they were when the greenhouses were warmed with kerosene heaters. The burning of the kerosene in the heaters actually produces a molecule similar to ethylene that synchronizes the ripening of the tomatoes. Now, the formation of ethylene requires oxygen, and the agricultural industry has used this information to their advantage. If you control the partial pressure of oxygen and carbon dioxide in a truck carrying any produce, you can prevent ethylene synthesis and thus slow the ripening process. This is helpful when fruits and vegetables are grown in one region of the world and then shipped many, many miles away to be sold. In the context of seed germination, ethylene is considered as a positive regulator. It stimulates seed germination and enhances germination rate of both immature and old seeds. It regulates auxin levels in dormant seeds and acts synergistically with GA and red light to induce germination in seeds, especially that of lettuce. Ethylene has been used to break dormancy in seeds of crops such as peanut and sunflower. In summary, aside from the effects of the genetics of the seed and environmental factors, seed germination is also regulated by chemical compounds known as phytohormones. Phytohormones are chemical signals that are produced at very low concentrations in the plant but affect all aspects of plant growth and development, including seed germination. The five major classes of plant hormones are abscisic acid, gibberellic acid, cytokinin, auxin, and ethylene. Each of these plant hormones regulates seed germination in various ways. Abscisic acid suppresses germination. Gibberellins or gibberellic acid promote germination. Cytokinin enhances seed germination by alleviating the effects of abiotic stresses. Auxins at higher concentration will inhibit germination but at lower concentration may promote or may not have any effects on germination. Ethylene positively stimulates seed germination and enhances the germination rate of both immature and old seeds.